The Innovation Postdoc Program at the University of Notre Dame is bridging the gap in the research and development process. This familiar, smooth-looking product pipeline is often not so smooth. There are obstacles creating a large gap between research and development, sometimes called the Valley of Death. While the basic research has been done, working concepts or prototypes need to be developed to bridge that gap. Scientists don't always have the resources to do this development work. Yet without working models, these projects aren't business plan ready. At Notre Dame, we see these problems as opportunities. But what's the best approach? Well, what if we could get help from someone who really understood practical innovation, say Thomas Edison? At Notre Dame, we do. Edison's son, Charles, set aside funds specifically to promote what his father was so good at, innovation. With support from the Charles Edison Fund, the College of Science has been able to bridge that gap by establishing the Edison Innovation Postdoc Program. The college is now matching these resources with existing funds, ensuring the program's viability for at least the next 2 years and supporting as many as 10 projects each year. It is hoped that eventually these projects themselves will show returns and support future innovations. A great example of how this works is a project dealing with colon cancer and individualized medicine, which relies on genetic test results. In spite of its potential impact, this project did not start in a lab. It started with a professor, a computer, and some extremely large data sets from the National Institutes of Health, known as microarray data. While studying this big data, mathematician Stephen Buechler and his collaborator, chemist Amanda Humman, made a significant discovery. Stage 1 and 2 colon cancer patients generally don't get chemotherapy following surgery. But while analyzing the microarray data, they discovered that some stage 2 patients could be identified genetically as having a much poorer prognosis than others in the same group. From an outcome standpoint, they really looked more like stage 3 patients. If this could be proven, then like stage 3 patients, they should also get chemotherapy. This they proved mathematically. So what's next? Buechler and Humman need to develop a test to identify this trait in future patients, and they've combined their talents to do just that. Those patients identified by the test could then have chemotherapy added to their treatment plan, and projections show that deaths for patients in this group could be reduced by 18%. Let's switch gears and talk about stroke, the third leading cause of death in the United States and the primary cause of long-term disability. There are 2 basic types of stroke, the ischemic or clotting type, which make up about 80% of cases, and the hemorrhagic or bleeding type, which comprise the other 20%. Currently, there's only one FDA-approved treatment for stroke in the United States, a protein-derived drug called TPA. It can only be used on the clotting type and must be used within no more than 4.5 hours of the stroke. TPA is a great clot buster, but it also promotes swelling and bleeding, exactly what's not helpful for the 20% of stroke victims who have the hemorrhagic type, where bleeding is the problem. So, once patients get to the hospital, they need a CT scan first to show there's a clot for TPA to work on. For most, by the time this testing is done, the treatment window is passed. Currently, only 3% of stroke patients are actually treated with TPA. Chemists Shahryar Mobasheri and Malin Chang have developed a compound called SB3CT, which has been shown to protect brain cells from dying regardless of the type of stroke. In fact, a water-soluble analog of this compound has been shown to reduce the damaged area of the brain by 60% in mice inflicted with stroke, with significantly reduced bleeding and swelling. By using it in combination with TPA, it is thought that the TPA treatment window can be widened to about 6 hours. With the basic research done, the optimal delivery mechanism still must be developed. The Innovation Postdoc Program has provided resources to do just that. Eventually, it is expected that these therapies may be administered by paramedics in the ambulance, giving immediate benefits. Now let's take a look at an example affecting global health. Each year, 8 million people contract an active form of tuberculosis, and 1.5 million people die from it, mostly in developing countries. There are a number of existing tests for TB, but all have significant drawbacks. One thing these tests have in common is that they require sputum, but AIDS patients, and many children, more than half, can't produce a sample. The standard smear test is very specific. If it says you've got it, you definitely do, but 50% of the time, when it says you don't have it, you still might. What's known as the culture test is much more sensitive, reducing false negative results, but requires incubation equipment and takes 2 to 4 weeks. Newer genetic tests require specialized equipment and are very expensive. So some people can't be tested at all, and when tests can be used, they are either not sensitive enough, require expertise in specialized equipment, or are very expensive. All of this adds up to one basic conclusion. Access to proper testing is severely limited, in particular for those who need it most, the populations of developing countries. Biologist Jeffrey Shorey is developing a test that uses a simple blood sample. This test will measure the mycobacterial components that come directly from tuberculosis, rather than measuring the patient's response to the disease. But finding these in blood is like finding a needle in a haystack. In order for this test to work, the samples must be enriched. One way to do this would be to run the blood sample through two membranes, one that filters out everything bigger than what you are looking for, and one that lets everything pass that is smaller. The resulting enriched sample could then be tested using a dipstick test, much like the familiar consumer pregnancy test. The Innovation Postdoc, working with Professor Shorey, is helping to develop these enrichment and detection processes. What does this mean? If successful, it means that everyone can be tested. It means that tests could be administered at the point of care, the doctor's office or clinic. It means the test will be affordable, and it means results in minutes, not weeks. It means that contagious patients can be identified immediately and kept from infecting others. The bottom line, it means availability for those in developing countries who have no access now. It is estimated that 400,000 lives could be saved each year if a point-of-care test like this can be developed. Notre Dame is making a concerted effort to invest in projects that have commercial potential, that have the very real possibility of translating into developed products that serve mankind. It's how we fight, with innovation.